Welcome back to Jalen Bio. Today's module is the blood system. In this session, we'll cover the following learning objectives: describe the general functions of blood, list the three components of centrifuge blood sample, name the three formed elements of blood and compare their relative abundance, describe the structure of erythrocytes, list the events by which erythrocyte production is stimulated, explain the main function of leukocytes, explain what is meant by differential count and how it is clinically useful, compare and contrast the different blood types and their importance when transfusing blood, explain vascular spasm, the first phase of hemostasis, and name conditions that bring about vascular spasm. Define coagulation and list the substances involved in coagulation. Compare and contrast the intrinsic pathway and the extrinsic pathway for activating blood clotting. Describe events in the common pathway. Alright, let's get started. Composition of blood. Blood transports oxygen and carbon dioxide to our lungs. It provides nutrients to the GI tract, it transports hormones from the endocrine glands, and heat and waste from systemic cells. It regulates our body temperature by absorbing additional heat from cells and releasing it through the surface of our blood vessel. It also regulates the pH by absorbing acids and bases from cells. The blood color depends on whether or not it's oxygen-rich or oxygen-poor. Oxygen-rich blood appears to be more bright red or scarlet in color, whereas oxygen-poor blood tends to appear more somewhat bluish. The blood volume is typically about 5 liters, but can range between 4 to 5 liters, maintaining blood pressure. Blood is 4 to 5 times more thicker or viscous than water, and it depends on how much solids are dissolved in the blood. The pH of blood ranges from 7.35 to about 7.45, and this is critical for preventing any kind of damage to proteins. So we take a sample of blood and we put it into a centrifuge machine, it spins it down, and we can collect the heavier elements at the bottom. The bottom layer of the centrifuge product contains erythrocytes, which makes about 44% of the blood sample. The middle layer is a layer called a buffy coat. This is made up of leukocytes and platelets. They form less than 1% of blood. The plasma, which sits on top of that layer, is a pale yellow liquid layer that is about 55%. So it ranges between 46 to 63%. You'll see that there are different kinds of form elements in there. There are the platelets, white blood cells, and red blood cells. And there's some categories of the leukocytes, which we'll talk about as well, like neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And notice that neutrophils dominate in 50 to 70% of all the white blood cell types. Some of the key proteins that we'll talk about include the albumin, which is 60% of the plasma protein inside the blood. Globulins come to about 35%, and fibrinogen, used for clotting into fibrin, is about 4%. Albumin plays an important role for maintaining osmotic pressure, filtration, and the absorption of fluids. Globulins are used for transportation of hormones and compounds, and fibrinogen is used for clotting. The components of the plasma proteins. Inside the plasma, we're going to have water, which is more than 90% of plasma by volume, helps maintain the temperature, and helps buffer and absorb any heat. And the remainder is going to be plasma protein. The blood or colloid consists of plasma proteins called albumin, globulin, fibrinogen. And there's also some regulatory proteins like enzymes, hormones inside the plasma. The albumin maintains the colloid osmotic pressure, also known as COP for short, colloid osmotic pressure, COP. That is a pressure exerted by the plasma proteins to draw the fluid into the blood. Therefore, prevents any kind of excess fluid built up between the capillaries and the interstitial fluid. We have the alpha globulins, which transport metal ions, lipids, whereas beta globulins transport lipids, and then the gamma globulins transport antibodies. There's the alpha globulins, there's the beta globulins, and there's the gamma globulins. Serum is plasma with the clotting proteins removed. Some of these regulatory proteins, the least abundant of the plasma protein, consist of enzymes that function to accelerate reactions as these hormones are transported around. 55% of the plasma is composed of the plasma protein, which is then divided into the albumins, globulins, fibrinogen, and other hormones. And the form elements is about 45%, which includes that buffy coat, which is less than 0.01% of the leukocytes. Thrombocytes is about 0.01%, and then 99.9% is all red blood cells or erythrocytes. A complete blood count provides a detailed panel about information of a person's blood sample. Here are some of the key metrics in a complete blood count analysis. WBC and RBC, these are the number of red blood cells and white blood cells per volume of blood analyzed. You'll see that the reference range is about 4.6 to 10.2. HEB is the mass of hemoglobin found inside a given sample of blood, and this can range between 12 to 16. HCT, which is hematocrit, is going to range between 37 to 47. That's the amount of the cell-like substances into the blood. MCV stands for the mean corpuscular volume, which is the approximate size of our red blood cell, and that can range between 77 to 99. The MCH, the mean cell hemoglobin, is the average amount of hemoglobin within each red blood cell, and that can range between 27 to 31. MCHC stands for mean corpuscular hemoglobin concentration, is the amount of hemoglobin in a packed volume of cells, and that ranges between 31 to 37. Last but not the least is RDW. This is the red blood cell distribution width, which approximates the fluctuation of the average amount of hemoglobin from cell to cell, and that ranges from 11 to about 14. And the last value, PLT, is the number of platelets of volume analyzed. Erythrocytes are the major formed element. Formed elements in the blood, approximately 45% of whole blood, is comprised of the formed elements, which are red blood cells, and platelets. Erythrocytes or red blood cells are these anucleated formed elements that transport oxygen and CO2 between tissues and the lung due to their very interesting biconcave shape that allows the maximal efficiency of movement of the oxygen around the bloodstream. A red pigmented protein consists of 4 protein building blocks, 2 alpha units and 2 beta units, and these are called the hemoglobin. Each globin chain contains a structure called a heme, which you'll see in a second. That heme is what is allowed to bind to the oxygen. These cells contain very little bit of DNA, and they're unable to synthesize protein. The process of erythropoiesis. Erythropoiesis begins with the stimulation of a myeloid stem cell by these multi-CSF signals, eventually forming what we call the proerythroblast. That proerythroblast then is an erythroblast that is producing hemoglobin. Then a smaller normal blast with more hemoglobin and has ejected its nucleus, and lastly becomes what we call the reticulocyte. You'll see right here the nucleus gets ejected in a critical phase and lasts about between 7 days to transition to reticulocytes. Similarly, white blood cells also go through a process called leukapheresis, which also involves granulocytes, monocytes, and lymphocyte maturation. And that process is very similar. It uses the myeloid stem cell to be stimulated to form into a progenitor cell, and does require some of these kind of key signals like the multi-CSF. These are the cell stimulating factors, and GM-CSF. These are important cell signaling factors that are used to help differentiate the cells as they mature. As it goes from the left to right, each globin chain contains a bigger amount of heme group, also known as hemoglobin, which contains iron, and that iron can then transport oxygen. As it gets bigger, the organelles inside the cell degrades, and the cell's nucleus also then gets smaller, eventually forming the mature erythrocyte. One clinical application is bone marrow transplant. This is a treatment for blood-borne cancers in combination with chemotherapy as well as immunosuppressive therapy. The bone marrow is obtained from an anesthetized donor through the iliac crest around the pelvic bone. Extremely painful. The donor is typically somebody that's related or close to the patient, and you can extract that bone marrow and then help replenish the blood cells to avoid any kind of permanent damage. This is a very grueling and painful process, as you can see in the image. Regulation of erythropoiesis by EPO. The kidneys are the primary producers of a hormone called erythropoietin. This is the key stimulator for erythrocyte production. It's stimulated by an oxygen level decrease in the blood, which is detected by our kidneys. As a result, this will then cause the red bone marrow to increase the production of erythrocytes, resulting in an increase of oxygenated erythrocytes and an increase in blood oxygen levels. This whole process is controlled by negative feedback. And other chemicals like testosterone can also stimulate the kidney's production of EPO. Therefore, those that have higher testosterone levels in males will generally have a higher erythrocyte count and also higher amount of hematocrit. So you'll see that when you have hypoxia or drop in oxygen, stimulates your liver and kidneys to begin to form EPO. EPO will then have several functions. It's going to target the bone marrow to begin the process of stimulating the proerythroblast to respond to those CSFs. And then the myeloid stem cells begin to differentiate, and you'll see the EPO receptors on hematopoietic stem cells. That will then eventually lead to the production and maturation for more red blood cells, therefore allowing you to have a higher binding capacity of oxygen. Blood doping is artificially inducing the amount of more blood cells by taking exogenous amounts of testosterone EPO packed in these really tight suspensions. This is considered illegal, and some athletes will use this to boost up their RBC levels. One method used is to donate your own blood to yourself. A few days prior to competition, a person might remove a certain amount of blood, allow time for the body to replenish that, and eventually inject it back into them to boost that up. Other methods include injecting the pharmaceutical grade EPO that can also boost directly the amount of oxygen. One application here is anemia, blood-based disorders characterized by the body's failure to supply oxygen to the tissue. Pernicious anemia is an example where the body fails to absorb vitamin B12 due to a lack of this protein called intrinsic factor inside the stomach. Other ones include iron deficiency anemia when you don't eat enough iron inside your diet. Hemoglobin reversibly binds to blood gases within the erythrocytes. So each of those globin chains contains a structure called a heme group. This heme group allows for binding of oxygen to the iron component of the heme group, and thus can transport in the blood. Because each of these hemoglobin molecules has 4 heme groups, it can bind up to 4 oxygen molecules through a weak bond that allows for rapid attachment and detachment of oxygen. CO2, carbon dioxide, also forms a relatively weak bond with the globin protein as it moves through the bloodstream. So it binds both compounds. Each chain contains a heme group that binds oxygen using iron. When it's bound to oxygen, we call that oxyhemoglobin. When it's not bound to oxygen, we call it deoxyhemoglobin. And the binding sites for oxygen are considered cooperative. So as you bind one, it increases the ability for the next one to bind. Let's talk a little bit about sickle cell anemia. Sickle cell anemia is an autosomal recessive anemia disorder in which a person inherits 2 copies of the sickle cell gene. As a result, the erythrocytes are going to be sickle-shaped and not the biconcave shape. This then lowers their ability to bind blood oxygen. As a result, there's a risk where the cells could be trapped in smaller blood vessels, causing clots. And this is considered one of the most heritable blood disorders of the list. So you'll see over here that instead of having a nice biconcave shape, we have a cross-section of a moon sickle shape. And as a result, in some of the smaller vessels, these regions could get clotted, causing problems. Erythrocyte destruction and processing. Erythrocytes lack a nucleus and organelles, yielding them unable to synthesize proteins that are necessary for repairing and making them extremely vulnerable to wear and tear. They last about 120 days, but all their components are recycled or excreted from the body. Macrophages located inside our spleen and our liver are able to phagocytize and break down some of the old red blood cells. Hemoglobin is also removed from the erythrocyte, with a heme group still attached to the globin protein. That globin is then broken down into free amino acids that can be recycled in the body. The heme groups does contain iron. Iron is Fe2+. At first, it's converted within the macrophage to this greenish pigment molecule called biliverdin. And the biliverdin gets converted into bilirubin, which is a yellow component found inside our digestive tract, inside the bile juices. And then that gets converted into urobilinogen, and then where it can be converted either into the stercobilin, which is going to be kind of a brown compound pigment that is going to be incorporated into our feces, or converted into urobilin, which is going to be excreted as urine, or urobilin in the urine. The iron components of hemoglobin are transported and removed by a protein called transferrin, TRF. Transferrin is going to then move it to the liver or spleen where it binds to ferritin, as well as another protein called hemosiderin. And these ferritin proteins are water-soluble, and they're primary storage sites for the storage of iron. On the left-hand side, you can see the processing of the red blood cell. Red blood cell gets broken down by macrophages into spleen, liver, and red bone marrow. It gets stripped into the heme group, as well as the globin. The globin is broken down into amino acids, where it can be recycled back into the bone. The heme group gets processed into biliverdin, that green compound. Biliverdin becomes bilirubin, and is transported inside the bloodstream, eventually creating into the stercobilin for fecal matter, the brown compound, or some of it can also be processed into urobilin, into the urine. The iron molecule is going to be transported with the protein called transferrin, and this will then bind onto the ferritin and hemosiderin inside the liver or spleen. And iron can also be recycled back into the bone marrow for processing and making more red blood cells.